Hello, Year 12. This is Mr. Isan. Thank you so far for your work with Chapter 17 and Photosynthesis. I got a little bit of feedback from some students who were finding some of this difficult last week's task, so I thought I'd give this video a go. Hopefully it will work, and I'll hopefully make things a little bit clearer for you. So this is a PowerPoint that I sent you last week, and it starts with a very simple photosynthesis equation here. Something we should recognize, nothing tricky about this at all. Couple of visual clues. All of this, most of the time, is happening on the leaf, so there's a diagram of a leaf there. Light energy is obviously involved, and to balance the equation out, we know that we need 6 molecules of carbon dioxide, 6 molecules of water, and with a bit of light energy, it will be converted to a molecule of glucose and 6 molecules of oxygen. Okay, so nothing too tricky there. Moving on to this slide here, which is an overview, and this is really quite a nice slide because it tells us, sort of summarizes the whole process. Okay, so in the past we've known photosynthesis perhaps as one reaction. It is one process. You get a bit of carbon dioxide, some water, and it turns into this and that. However, at A level, we delve a little bit deeper into it and find that actually photosynthesis is a combination of 2 main reactions. Okay, one which we call the light-dependent reaction, or the light reactions, and the second one which is the light-independent stage or reaction, and that is also known as a Calvin cycle. Sometimes it's called the dark reactions. Okay, but it's now most commonly known as the Calvin cycle, developed by Melvin Calvin. And both of these are interlinked. So the light reactions depend on the Calvin cycle and vice versa. And as you can see from that diagram there, in the light reaction we're making 2 things. Okay, namely ATP, which is going to provide the energy for the reactions in the Calvin cycle, and NADPH, or reduced NADP. Now both of these things are needed for the Calvin cycle to work, and the Calvin cycle uses the phosphate from the ATP, converts it back to ADP and an inorganic phosphate, and sends it back to the light reaction. And also the NADP is then oxidized and sent back to the light reactions, where once again it is reduced, the ATP is generated, phosphorylated, and heads back here. So both of these are interlinked. Also looking at where these raw materials are used. So we know that light reactions are where the water is used and the oxygen is produced. And the Calvin cycle is where the carbon dioxide comes in. So even though carbon dioxide is an essential raw material for photosynthesis, it doesn't get involved until the second stage, the Calvin cycle. And the Calvin cycle is also where the more useful organic molecules like sugars and fats and proteins can be generated, okay, through this process. So let's have a look where this is happening. This is taking place obviously in the chloroplasts of plant cells, and you should have a good idea of the structure of chloroplasts from the earlier chapters in AS, or the year 1, and you'll find that there are stacks of thylakoids. Within those thylakoids, on the membranes, is where the light dependent stage takes place. Okay, the light dependent stage. The light independent stage, which we'll check a little bit later, is happening in the stroma. That's the area surrounding the thylakoid stacks. Okay, so the light dependent stage is where we go first. Looking at that a little bit closer, we find that actually within the membranes of these thylakoid stacks you have photosystems. Photosystems are like a funnel-shaped structure where you have a reaction center at the bottom. So any light falling onto the top of these photosystems is sort of absorbed and harnessed by a mixture of different pigments, such as chlorophyll B, xanthophylls, carotenoids, all of these funnel the light energy down to the reaction center where you have your primary pigment, which is chlorophyll A. The others are known as accessory pigments. They sort of support chlorophyll A in harvesting this energy. So they funnel this down to the reaction center where the energy is used to carry out the process. Okay, and obviously these photosystems are in the membranes. The membranes are within chloroplasts, and that is usually found on the leaves. Okay, so here is the light dependent stage as a process. Now on first glance it looks rather complicated, and it's not too easy to get your head around. We'll give it a go, and hopefully the more you do it, the easier it becomes to understand exactly what's going on at each of these steps. Okay, so just quickly, I'll do the mistake thing first. This arrow should be pointing the other way, of course. NADP is reduced by this enzyme, and then the arrow should be pointing that way to say that it is going to go after the Calvin cycle. Anyway, we'll ignore that for a minute, and we'll carry on with what this process is. So here we've got two photosystems, something we talked about just a short while ago. There's photosystem 2 and there is photosystem 1. Now the first thing to sort of get your head around is why in the world is photosystem 2 written first and photosystem 1 later on. It would make sense obviously to have this one here, photosystem 1, and this one to be photosystem 2. It is only a matter of when they were discovered. So photosystem 1 was discovered first, they named it photosystem 1. And they studied it and they found that it does all this stuff here. And then they found another photosystem which was involved, and they named it photosystem 2 and realized that actually it gets involved a little bit earlier in this process. So that's all it is, nothing else is going on with that. Okay, so you've got light. Now when light falls onto these photosystems and onto the leaf, it's doing three things. It's falling on photosystem 1, it is falling on photosystem 2, and it is falling also on the water which is within the plant cell there, within the chloroplast, and it carries out what we call photolysis, or photolysis if you were to break that word down, which effectively just means breakdown, lysis, because of light, because of photo. So photolysis is really the first place to start here. All it means, as we said, is that the water is breaking down into its constituent parts, which are two protons, H pluses, which have come from the H2 bit, half an oxygen molecule, because it's not quite an O2, it is half an O2 because it's just one O, and to make up the rest of the hydrogens, we also had two electrons that were produced. Okay, so these three things are produced in photolysis, and at this point we are going to ignore the oxygen because we are going to consider that a bit of a waste for this process, and this is the only time when oxygen gets involved. Oxygen leaves from here, perhaps gets involved in a bit of respiration, leaves through the stomata, and is useful for us, but for the plant, for this process, it plays no further part. The protons here, the hydrogen ions, will get involved later on, so I'll talk about those in a short while. What I'm going to focus on for now is the electrons that were produced from photolysis. Photosystem 2 here, remember, was also struck by light. Now, when it's struck by light, what we're saying is that little pockets of energy, what we call photons, hit the photosystem. The energy is harnessed by the photosystems and it funnels down, as I said a short while ago. Now at this point, two electrons are lost from chlorophyll molecules. So chlorophyll molecules contain magnesium, and within that magnesium atom, you lose two electrons. Why do you lose these electrons? Because what effectively happens is the light gives those molecules so much energy that the kinetic energy effectively separates them from the molecule, and they sort of are raised to a higher level. Okay, as you can see depicted by this arrow here as well. And when these electrons are raised to a higher energy level, they've only got really one way to go. They are picked up by electron carriers, okay, and every time they are passed from one electron carrier to the next, they lose a little bit of their energy and eventually end up in photosystem 1. And I'll just pause there for a second, I'll come back to that in a second. What happened here, the 2 electrons that were lost, were actually then compensated by the 2 electrons from the water. They join photosystem 2, and photosystem 2 is now set and ready to go again if needed. These 2 electrons were here. When the light hit photosystem 2 and water, it also hit photosystem 1. And that lost electrons in the same way as photosystem 2 did earlier. So those electrons are up here. Now these electrons will take that space, having travelled through the electron carrier system. Now this here is quite a complicated process in itself, which we'll talk about later. And it's really simple here, a straight line going to PS1, but actually there's a lot going on here which effectively generates some ATP. We'll talk about that later on. But sticking with this for now, the 2 electrons are released here. Now at this point is a bit of a junction where the electrons could either carry on through the process of non-cyclic photophosphorylation if some NADP is present, or if there is no NADP, they will actually go back into the electron carrier system, and this is where things get a little bit confusing, because these electrons which were going through the system and these electrons going through the system, they're both going to fight to get onto photosystem 1, and there's only space for 2 electrons. So what tends to happen here is that these electrons can't really go any further, and they drop back down to PS2. The electrons from the water are not needed, and the process effectively stops. You do get a little bit of cyclic photophosphorylation because these electrons keep getting energized, excited, then drop back down, and as they do, they generate a little bit of ATP. So ATP production does carry on in some small way through cyclic photophosphorylation, and it's obviously cyclic because it's kind of going round and round. But in the plant's interest, non-cyclic photophosphorylation is the way to go. So it really wants some NADP here, which can be converted to NADP, reduced NADP, via NADP reductase, which is an enzyme. So if those 2 electrons are available, what tends to happen is those electrons are picked up along with the protons. Remember them from a little while ago. Along with the protons, and these electrons and those protons make up 2 hydrogen atoms. Now these 2 hydrogens will be added to 2 NADP molecules, leaving you with 2 reduced NADP molecules, otherwise known as NADPH. So I said earlier on that for the process of photosynthesis and the Calvin cycle, we need some reduced NADP that goes from the light-dependent stage to the Calvin cycle, and some ATP, which goes from the light-dependent stage to the Calvin cycle. At this point, we have managed to produce both of those. We've got some reduced NADP, and we've got some ATP. So, actually, we're ready to go into the Calvin cycle. Okay. So if you want, you can sort of pause it there and go back, listen to that again, and try to understand what is going on at each of these stages. I don't think I've missed anything off. As I said, this bit here needs a little bit more explanation, but I'll do that in a separate video so as not to sort of congest this one too much. Okay, so that's the light dependent stage. Now this on the PowerPoint, there's quite a bit of information here, hopefully similar to what I've just said to you, so you can refer back to this as well and see if it all makes sense. This here is that electron carrier system, and as I said, I will talk to you about that in a different video. Then there were some questions. Now you could practice this question. Hopefully you managed to have a go at this. If you did and didn't feel really comfortable with it, having listened to the video, try and give it another go, and you can mark it yourself using this mark scheme. Now the light independent stage, or the Calvin cycle, is where we're heading next. Where did I say this happened? It happens in the stroma. Now the stroma is just a space in between these thylakoid stacks. It is part of a chloroplast, which is part of a plant cell, and that, of course, is in the leaf. So we go further and look at the Calvin cycle. This is it here. Now remember what we brought over from the first stage. It was reduced NADP, which is going to get involved in various different steps here, convert back to NADP, and ATP, which itself is going to get involved, provide the energy. We know ATP is the energy currency for living things, so ATP is going to provide the energy for some of these processes, and once it becomes ATP and the reduced NADP becomes NADP, they will head back to the light dependent stage, and that process will carry on. Okay, so what's going on here in the Calvin cycle? Calvin cycle involves this enzyme called ribisco. Its full name is ribulose bisphosphate carboxylase oxygenase. Okay, long old name, so thankfully they've shortened it to ribisco. It's got a nice ring to it, and it is immensely important for living things. Okay, without this enzyme, of course, photosynthesis wouldn't make organic molecules, and those organic molecules then wouldn't be eaten by us animals, and life as we know it wouldn't exist. Okay, so ribisco is vitally important. What does it do? It is involved in the process of carbon fixation. Now carbon fixation quite simply just means getting carbon dioxide in its gaseous atmospheric form and turning it into hard and fast molecules, chemicals, which can then be incorporated into make bigger ones, which are useful for organisms, plants and animals alike. So how does it do that? It does that by having an active site which combines something called RUBP, or ribulose bisphosphate, which is a 5-carbon compound, with a carbon dioxide molecule. Once it does that, the 5 plus the 1 carbon from here allows it to make a 6-carbon compound, which quite quickly is broken down into 2 3-carbon compounds called glycerate 3-phosphate, or GP as it's known. Now that process here is carried out by ribisco. After this, the GP molecules are converted to triose phosphate molecules, or TP. Okay, for this, hydrogens are needed. Where do the hydrogens come from? They come from the reduced NADP, whose basically the sole job is to carry hydrogen from one place to another and give it where it's needed and pick it up so that it can replenish its sort of supplies. And it brings those hydrogen supplies and uses them here to make TP. This is also an energy consuming process, so ATP through its dephosphorylation provides the energy for it, and TP are made. Now it's worth noting at this point that even at this early stage of the Calvin cycle, we can start to produce some fatty acids and amino acids to be used by the plant. Okay. Now, at this point, the TP is where the plant starts to see some of the fruits of all the work that it's put in so far. Okay, so all the photolysis and phosphorylations and Calvin cycles, this is where it sort of bears fruit. Because the TP can be converted to hexose sugars like glucoses, they can be converted to sucrose, cellulose for cell walls, fructose in the fruit, starch to store, lots of different things. It can also be converted to lipids via glycerol and some of the fatty acids that were produced earlier on, and lipids can be formed. Amino acids can be used to make proteins. And this is where the last part of the equation, where we kind of really summarize it and say glucose, that is actually what we mean when we say glucose. We actually mean a whole host of organic molecules. Okay. Now at this point, every 5 out of 6 TP molecules are actually regenerating the IUBP, which again is an ATP requiring, energy requiring process. Only 1 out of those 6 is used to make these things. Okay, now that's quite a useful thing because it makes sure that this cycle continues to go on. If too much of that was used to make these molecules, the efficiency would be reduced. So in the interest of efficiency, lots of this TP is actually goes on to regenerate IUBP, and a small amount, 1/6, is used to make these useful molecules. Now the IUBP here is going to go back and fix some more carbon with the help of Rubisco. Our two friends from the light dependent stage, ATP, will now become ADP, and reduced NADP, which has now become NADP, can head back and start to get involved with this process again. Okay, they can then be reduced, ATP can be generated from ADP, and they can head back to the carbons. If you have any further problems, what I'll also do is I will make another video about the electron transport system, which helps to generate the ATP. Okay, thank you for listening.